So good afternoon, everyone. Today I'm going to tell you a little bit about what my lab works on, and actually just two thrusts of a couple more. And one of them is in developing biomimetic materials, and the other is making nanoscale materials for the delivery of nucleic acids. So I'm going to start with the biomimetic materials. So like our first speaker, we also take some inspiration from nature, however, on a completely different length scale. So our motivation currently are these structures called biomolecular condensates. They're also known as membraneless organelles. These are structures that form within cells, and they are liquid droplets that are dynamic, and they can form and dissipate. They have a variety of different functions, kind of a hot topic in biology. Because until about 10, 15 years ago, people didn't realize they were liquids. That's mainly because of our limitations in imaging within the cell. Now that our imaging techniques have become much better, people can see these droplets form within cells, and then they coalesce, and then they relax, exhibiting the properties of a liquid. And so these droplets, they're made of proteins and nucleic acids. And some of them can have multi-layered structures, and all of these structures, all of these layers, are liquids. So I'm going to show you a video here. This is a structure that our lab is really interested in recently. It's called the nucleolus. This particular image is taken from this publication here. It's the nucleolus in frog eggs. But as you can see, right, you can see these things fused. You can tell they're liquids. And in this particular structure, you can see how there's a green center and a red exterior. These are two different liquid phases. There's actually a third liquid phase that's smaller, and it's not labeled here, so you can't see it. But the structure of this nucleolus is amazing. Its function is to assemble the ribosome. And what it does is it assembles the structure from the inside out. So each different liquid layer has its own components. It's able to exclude what it doesn't need. It entraps the enzymes it needs for its function, and then it passes it on to the next liquid layer. Similarly, that layer has only the molecules it needs, and it excludes what it doesn't, and so forth for the third layer. So when I look at that, I'm a chemical engineer by training, and I see a very, very amazing chemical reactor that can exclude what it doesn't need, only uses what it needs. It can form, and sometimes it can fall apart when it's done. I think it's quite remarkable. In the biological communities, people have been identifying the proteins that form these structures, right? They're made of proteins and nucleic acids. What people know is that the proteins are what we call intrinsically disordered proteins. So they don't have a fixed structure. Their structure is fluctuating all the time. These intrinsically disordered proteins, or sometimes regions within a protein, have sequences, so series of amino acids that are really low complexity and very repetitive. So very different from a normal protein structure. And some of the well-studied examples of these proteins suggest that formation of these liquids involves mixing of oppositely charged molecules. So things like electrostatic interactions or aromatic interactions seem to play a role in the formation of these droplets. So my lab works with peptide-based materials. And so when we looked at this problem, we thought, okay, proteins are really complicated. We can use peptides to try to understand fundamentally how you can form these liquid droplets. Because peptides are easy to design and engineer. They have a lot less complexity than proteins. We can isolate different molecular interactions. So one of the goals of this project is to understand fundamentally how to form these droplets, and what makes them stable, how to maybe make them fall apart. And the other goal is to actually recreate this multi-layered structure and put enzymes in it and do some biocatalysis. I'll talk about the first goal first. We've I'm not going to go into all the details, but we've made basically a small library of different peptides that can form these liquid droplets, or that we thought could form these liquid droplets in some cases. And so we're just making oppositely charged polymers, so polycations and polyanions. They have very simple sequences. Sometimes we have less charge, sometimes we have more charge. We're looking at different amino acid incorporation. That's what the X is in these sequences. X can be one of 5 amino acids. We're increasing in hydrophobicity from left to right. And then when we incorporate these ring structures, we're incorporating aromatic interactions as well. So we've made different versions of these, different iterations for the positively charged and the negatively charged molecules, with using all these different hydrophobic molecules with aromatic interactions. So we design them in the lab, we synthesize them, we do all sorts of characterization to make sure we have the correct molecules, and then we start mixing them together to see if we can form these liquid droplets. In a lot of instances, we can't. We form these solids. And so these solids, when we look at them using things like FTIR or infrared spectroscopy, we see that they're hydrogen bonded. And sometimes, though, we do form liquids. For our second generation of molecules, we formed a lot more liquids. And so we've kind of learned how to tune the interactions to the right level to form a liquid and not a solid. We've also learned a lot about how you can transition a liquid to a solid, which is actually something that's very prevalent in disease. Things like ALS and frontal temporal dementia involve kind of dysregulation of the membraneless organelles to form solids. So we're kind of have a spin-off project that's working a lot about that recently. So now that we've made these different droplets, they have different flavors. We're doing things like measuring the interfacial tension, and we start mixing them together to see if we can start making this multi-layered structure. And we've been able to do that. We've got more hydrophilic, more hydrophobic droplets, and we can make them form these multi-layers. We're starting to put enzymes inside them and see how they can work in these crowded environments, which are more similar to how enzymes work naturally. So that's kind of where we are now. We're constructing this, and we can encapsulate different molecules in the different phases. And so we think this has a lot of promise for biocatalysis, and we've learned a lot about what can make the materials. The second project I want to talk about has to do with nucleic acid delivery. So I'm sure we're all aware nucleic acids have a lot of potential as therapeutics, as vaccines, right? We all know the COVID vaccines, of course, were delivering messenger RNA, but you can't just inject RNA into the body. You need some sort of protective vehicle. For the COVID vaccines, this was a lipid nanoparticle. It's a great platform, but it relies on this, synthetic lipid that's quite cumbersome to synthesize. This platform I'm talking about is an alternative. I'm not saying it's necessarily better, but it's less studied. And so we've been working with this platform as a nucleic acid delivery vehicle. It's a very simple platform. Again, we're mixing oppositely charged polymers to form our nanoparticles. Except now, instead of just having a charged polymer, we also link to it a neutral block. So we call this a block copolymer. And in doing so, instead of forming these bulk materials that you can see sometimes with your naked eye, more likely with a microscope, you can form these things on the nanoscale. So you'll have a polyelectrolyte complex core surrounded by this neutral polymer. Sometimes you can decorate these things with targeting peptides. And in the past, we've done that for applications in heart disease. And you can see those references if you're interested in that particularly. You can design these types of nanoparticles to be spherical or worm-like. Those obviously have different implications in nanomedicine. But recently, what we've been trying to do is two things, and there's a lot of different factors that have influenced the morphology of these. I'm not going to go into that detail today, but what we've been trying to work on over the last couple of years is essentially making the exterior of these little nanoparticles responsive to temperature so that you can use that as an aid in either suppressing or promoting delivery of different molecules. We did projects with that with a PhD student that recently graduated. He did a lot of work in advanced characterization of these nanoscale materials. So we made, worm-like micelles that are more cylindrical and spherical particles. We characterized these at the Advanced Photon Source at Argonne National Lab with the aid of Xiaobing Suo. And we also looked at what happens with the structure of these micelles under temperature. So, you can actually see the worms lose their structure using a small angle X-ray scattering, while the spheres actually retain it. So we have different kind of avenues that we can take when using these for drug delivery. One of the things we're looking at now too is kind of re-engineering. That was kind of a re-engineering of the exterior of the corona. That's what we call it. But the interior is also engineerable. And so we're changing the interior from a liquid to a solid and seeing how that influences the release of molecules and the exchange of molecules between these types of assemblies. So with that, I think I've almost run out of time. I'd like to thank my current group members, Tahora, Akash, and Lindsay, who've are in the lab working right now, and alumni that contributed to this presentation. Notably, Sarah Tabende did a lot of the work with the Biomimetic molecular condensates, and Sachit Shah did a lot of the work with the polyelectrolyte complex micelles for nucleic acid delivery. And of course, our funding through NSF Career Award and 3M as well. And thank you, and I'd happily take any questions.